Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics. Most people think cancer cells die in one of two major ways: controlled programmed cell death called apoptosis, or uncontrolled cell death called necrosis. These are the classical pathways most people learn about, and many cancer therapies are designed, at least in part, to trigger them. But cancer cells are very good at resisting death. So researchers have become increasingly interested in another form of regulated cell death called ferroptosis. And ferroptosis is different. It's not just about intrinsic or extrinsic apoptotic triggers, caspase, cascades, or the apoptosome. It is about pushing the cancer cells into an oxidative stress crisis it can no longer buffer. Ferroptosis depends on a dangerous combination: iron, fatty acids, lipid peroxidation, oxidative stress, and the weakened antioxidant defenses. That matters because cancer cells often live close to the edge of an oxidative cliff. To survive, they rely heavily on systems like glutathione, glutathione peroxidase, thyroredoxin, NADPH, glucose, and glutamine metabolism. So the question is not simply, can we kill cancer cells? The better question is, can we make cancer cells ferroptosis sensitive and then push them over the edge? Today, we're going to break down ferroptosis, how iron, hoofa fats, glucose, glutamine, glutathione, glutathione peroxidase, and oxidative stress all connect, and why this pathway may be one of the most important redox vulnerabilities in cancer metabolism. So without further ado, let's get into it. So let's get into what ferroptosis is and how this is relevant in the fight against cancer. I'm going to start out with a paper that was published in March of 2024, and it was titled Ferroptosis in Cancer: From Molecular Mechanisms to Therapeutic Strategies. And it says here in the abstract, ferroptosis is a non-apoptotic form of regulated cell death characterized by the lethal accumulation of iron-dependent membrane-localized lipid peroxides. It acts as an innate tumor suppressor mechanism and participates in the biological processes of tumors. Intriguingly, mesenchymal and dedifferentiated cancer cells, which are usually resistant to apoptosis and traditional therapies, are exquisitely vulnerable to ferroptosis, further underscoring its potential as a treatment approach for cancers, especially for refractory cancers. However, the impact of ferroptosis on cancer cells extends beyond its direct cytotoxic effect on tumor cells. Ferroptosis induction not only inhibits cancer cells, but also promotes cancer development due to its potential negative impact on anti-cancer immunity. Thus, a comprehensive understanding of the role of ferroptosis in cancer is crucial for the successful translation of ferroptosis therapy from a laboratory to clinical applications. In this review, we provide an overview of the recent advancements in understanding ferroptosis in cancer, covering molecular mechanisms, biological functions, regulatory pathways, and interactions within the tumor microenvironment. We also summarize the potential applications of ferroptosis induction in immunotherapy, radiotherapy, and systemic therapy, as well as ferroptosis inhibition for cancer treatment in various conditions. We finally discuss ferroptosis markers, the current challenges, and future directions of ferroptosis in the treatment of cancer. And we're going to just introduce the topic with this paper and get into a little bit more of the nitty-gritty in the next paper. So in the introduction, it says, every living being eventually dies. Cell death is a key biologic process inherent in complex organisms, serving as a crucial mechanism for the elimination of unwanted cells. Mammalian cell death encompasses accidental cell death, an uncontrolled biologic event triggered by an unexpected attacks or injuries, and regulated cell death, which is driven by a genetically encoded apparatus and can be modulated by drug or genetic interventions. The orderly progression of regulated cell death in complex organisms is integral to its normal development and homeostasis, while the loss of controlled cell death contributes to human disease, such as cancers, characterized by the presence of abnormal cells exhibiting unlimited replication and immortality due to successful evasion of cell death regulation. Cancer treatment strategies consistently prioritize the selective eradication of cancer cells while minimizing harm to normal cells. That's arguable whether that's true or not. I'm sure that's the goal, but does that actually happen in clinical practice? Story for another day. Regulated cell death is an important channel for achieving this, as it enables the specific targeting of tumor cells and enhances the efficacy of drug-induced cell death while simultaneously reducing adverse effects on normal cells. Ferroptosis A term coined by the laboratory of Brent Stockwell in 2012 is a distinct mode of regulated cell death characterized by the iron-dependent lethal accumulation of membrane-localized lipid peroxides. Cells undergoing ferroptosis display distinct hallmarks compared to other extensively studied forms of regulated cell death, such as apoptosis, pyroptosis, and necroptosis. Morphologically, ferroptotic cells exhibit dysmorphic small mitochondria with condensed membranes and decreased cristae. Mechanistically, unlike classical regulated cell death involving specific executioner proteins of cell death, such as the gasdermin D for pyroptosis, caspase for apoptosis, and mixed lineage kinase domain-like proteins MLKL for necrosis. The identity of the cell death executioner proteins in ferroptosis remain unclear. While it is widely accepted that the execution for ferroptosis necessitates the oxidized phospholipids containing polyunsaturated fatty acids, PUFA, the mechanism by which these oxidized PUFAs beyond a certain threshold lead membrane permeabilization and cell death, as well as downstream executioners that mediate the eventual execution event known as the point of no return in ferroptosis, remain largely elusive. The process of ferroptosis involves ferrous iron accumulation, free radical production, antioxidant system dysfunction, and lipid peroxidation. Based on its distinctive features, a comprehensive panel of markers and functional tests, including pharmacologic inhibition, has been assembled to effectively differentiate ferroptosis from other types of regulated cell death, providing suitable tools for investigating and pathophysiological functions of ferroptosis. And I like this paper. It really gave us the history of how this happened. So they discovered there was an iron-induced toxicity back in 1908, but it wasn't until they started discovering some of these like XC, which is SLC711, a cysteine glutamate antiporter, glutathione peroxidase 4, aristin, RSL3, until 2012, the term ferroptosis was not actually coined, which is why it seems so novel for many of us who are in the biological sciences. So we're going to use this paper that was published in July of 2022 to really hammer down the key components of what makes ferroptosis special. The paper was titled Targeting Ferroptosis as a Vulnerability in Cancer. And it says here, ferroptosis is an iron-dependent form of regulated cell death that is triggered by the toxic buildup of lipid peroxides on cellular membranes. In recent years, ferroptosis has garnered enormous interest in cancer research communities, partly because it is a unique cell death modality that is mechanistically and morphologically different from other forms of cell death, such as apoptosis, and therefore holds great potential for cancer therapy. In this review, we summarize the current understanding of ferroptosis inducing and ferroptosis defense mechanisms, dissecting the roles and mechanisms of ferroptosis in tumor suppression and tumor immunity, conceptualize the diverse vulnerabilities in cancer cells to ferroptosis, and explore therapeutic strategies for targeting ferroptosis in cancer. And I'm going to skip down to where it starts talking about the components that are required for ferroptosis to actually happen. So this part of the paper is titled Ferroptosis Prerequisites. The crux of ferroptosis execution is iron catalyzed peroxidation or oxidation of fatty acids of polyunsaturated fatty acids, PUFA containing phospholipids, PLs, which, when exceeding the buffering capacity of the ferroptosis defense systems, see in next section, can lead to lethal accumulation of lipid peroxides on cellular membranes and subsequent membrane rupture, resulting in ferroptotic cell death. As outlined in this section, PUFA-PL synthesis and peroxidation, iron metabolism, and mitochondrial metabolism constitute the main prerequisite driving ferroptosis. So let's start out with the PUFA-PL synthesis and peroxidation. Acyl coenzyme A, CoA, synthase, long chain family member 4, ACSL4, and lysophosphatidylcholine acetyltransferase 3, LPCAT3, are crucial mediators of PUFA-PL synthesis. ACSL4 catalyzes the ligation of free PUFA, such as arachidonic acids and adrenic acids, with CoA to generate PUFA-CoA, such as arachidonic acid CoA or adrenic acid CoA, which are subsequently reesterified and incorporated into phospholipids by these other enzymes, LPCAT3, to form PUFA-PLs, such as arachidonic acid phosphatidylethylamine, PE, or adrenic acid, PE. Acetyl-CoA carboxylase, ACC, catalyzed carboxylation of acetyl-CoA generates malonyl-CoA, which is required for the synthesis of some PUFAs, like the elongation steps, and therefore for ferroptosis. Inactivation of ACSL4, LPCat3, or ACC blocks or attenuates ferroptosis. Interestingly, p53-mediated ferroptosis in some contexts seems to be independent of ACLS4, although how ferroptosis occurs in the absence of ACLS4-mediated PUFA-PL synthesis remains unclear. Recent findings also revealed that PUFA-containing ether PLs synthesized in peroxisomes act as additional substrate for lipid peroxidation, favoring ferroptosis onset. PUFA-PLs are particularly susceptible to peroxidation due to presence of bis-allylic moieties in PUFAs. The peroxidation of PUFA-PLs is primarily catalyzed by non-enzymatic auto-oxidation driven by the Fenton reaction with iron as the catalyst. Cytochrome P450 oxoreductase POR and ALOX-mediated enzymatic reactions have been shown to promote lipid peroxidation, although the ability for POR to promote lipid peroxidation appears to be indirect via the generation of hydrogen peroxide, and the role of ALOX in ferroptosis has been challenged by other studies, including the lack of rescue of glutathione peroxidase 4, GPX4, knockout phenotypes in mice. Monosaturated fatty acids, MUFA, such as oleic acid or palmitoleic acid, are not readily peroxidized owing to their lack of bis-allylic moieties. In contrast to PUFAs, MUFAs restrain lipid peroxidation and ferroptosis by displacing PUFAs from phospholipids in cellular membranes, and activation of enzymes involved in MUFA-PL synthesis, such as steryl-CoA desaturase 1, SCD1, and ACSL3, sensitizes cancer cells to ferroptosis. So that was a lot of jargon and enzymes and big giant words. But basically what the takeaway from this section is that monounsaturated fats and saturated fats are less likely to enhance or increase the likelihood of ferroptosis, whereas polyunsaturated fatty acids, because of all the double bonds they have, make it more likely to generate ferroptosis. So PUFAs, or polyunsaturated fatty acids, are at least one of the prerequisites to having ferroptosis happen more readily. Next prerequisites: iron metabolism. Iron governs ferroptosis not only by initiating the non-enzymatic Fenton reaction for direct peroxidation of these polyunsaturated fatty acids, PUFAs, but also by acting as an essential cofactor for enzymes that participate in lipid peroxidation, such as ALOX or POR. Cells normally maintain a reliable, stable pool of labile iron through orchestrated regulation of iron uptake, utilization, storage, and export. In balance, regulation of these iron metabolism processes can promote or suppress ferroptosis, depending on whether the level of intracellular labile iron pool is increased or decreased, respectively. For example, intracellular iron is stored mostly as inert iron in ferritin, whereas autophagic degradation of ferritin, ferritinophagy, releases stored iron in ferritin into the labile iron pool. Consequently, blockade of the nuclear receptor coactivator 4, NCOA4, mediated ferritinophagy decreases the level of labile iron pool and suppresses ferroptosis. Conversely, enhancement of ferritinophagy by inhibiting cytosolic glutamate oxaloacetate transaminase 1, GOT1, increases the labile iron pool and promotes ferroptosis, although exactly how GOT1 inhibition promotes ferritinophagy-mediated labile iron release remains to be further studied. We refer readers to the recent excellent review for detailed discussions on iron metabolism and ferroptosis. The next prerequisite deals with mitochondrial metabolism. Several metabolic processes in mitochondria have important roles in triggering ferroptosis. First, the generation of mitochondrial ROS, reactive oxygen species, is critical for lipid peroxidation and ferroptosis onset. Mitochondria are a major source of cellular ROS, and in which electron leakage from electron transport chain complexes 1 and 3 generate superoxides, followed by their conversion to hydrogen peroxide by superoxide dismutase. Hydrogen peroxide can then react with labile iron via the Fenton reaction to generate hydroxyl radicals, which subsequently drive PUFA-PL peroxidation. Moreover, electron transport and proton pumping in mitochondria are important for ATP production, which also promotes ferroptosis. Specifically under ATP depleted conditions, AMP-activated protein kinase AMPK phosphorylates and inactivates ACC, thereby suppressing PUFA-PL synthesis and blocking ferroptosis. In contrast, with sufficient energy, ATP, AMPK can be activated efficiently and ACC activated, promoting PUFA-PL synthesis and ferroptosis. Finally, the role of mitochondria in biosynthetic pathways in cellular metabolism contributes to ferroptosis. Ferroptosis requires the TCA cycle and various anapolaritic reactions that fuel the TCA cycle, such as glutaminolysis in mitochondria, which drive ferroptosis through promoting ROS, ATP, and/or PUFA-PL generation. Therefore, a multifaceted functions in mitochondria in bioenergetics, biosynthesis, and ROS, or reactive oxygen species generation, drive mitochondrial lipid peroxidation and ferroptosis. And then, of course, there is ferroptosis defense mechanisms that need to be impaired. And that's what it's talking about here. Ferroptosis defense mechanisms involve cellular antioxidant systems that directly neutralize lipid peroxides. Studies of ferroptosis defense mechanisms have been one of the most exciting and rapidly evolving areas of ferroptosis research in the past 2 years. As discussed below, there are at least 4 ferroptosis defense systems with distinctive subcellular localizations. And it had a pretty decent graphic that I pulled as part of this. And since we pretty much went over all of these, I'd like to just make this graphic bigger so we can actually look at this together and really kind of study it. So here are the kind of prerequisites, right, that are leading to the driving of lipid peroxidation and ferroptosis: PUFA fatty acid synthesis and/or availability, iron metabolism, specifically labile iron pools that are highly reactive with the hydrogen peroxide that are generated by the mitochondrial metabolism, which leads to lipid peroxidation, membrane damage, and ferroptosis. And what's interesting is there are a whole bunch of endogenous antioxidant systems, some of which we have covered in the past, like glutathione, glutathione peroxidase, CoQ10, but there's some other ones that have not been covered that are also critical at restricting lipid peroxidation and preventing against ferroptosis. And this graphic here shows how they kind of fit together in the cellular milieu, where glutathione, glutathione peroxidase are help protecting the mitochondrial membranes, and other antioxidant systems through the XC or the SLC711 glutathione system are helping protect other outside the mitochondrial membranes to protect against ferroptosis. And so when we look at how metabolic therapies are employed, it helps us understand activating ferroptosis specifically within cancer cells is a major goal of ours. It gives us specific targets of where we're going to be trying to modulate these systems in order to set these cancer cells up for being maximally ferroptosis sensitive prior to oxidative therapeutic interventions. And I'm going to go back to this paper that we started with because it had some excellent graphics. So it gives slightly more advanced graphics on how this is actually happening. Here we have the import of iron, which is happening at a more increased rate and amount compared to normal cells. Here's the LIP, labile iron pool. We see that interacting with the hydrogen peroxide generated by mitochondrial metabolism to create this Fenton reaction, which leads to the hydroxyl radical and oxidized iron. And then the hydroxyl radical, which is a more dangerous radical, is then interacting with these PUFAs in these membranes, which leads to membrane destruction and ferroptosis. And then you'll also see in the same graphic where XC or 7811, glutathione, glutathione peroxidase are helping to try to buffer these systems. And then it had a nice little graphic also towards the end of the paper where it combined how certain systemic drugs such as sulfasalazine, artesunate, statins, BSO, and other compounds are able to be combined in meaningful strategic ways to enhance things like radiotherapy or other oxidative stress inducing therapies. This is a critical video for us to understand what ferroptosis is so that we know what levers need to be pulled in order to maximize its occurrence within cancer cells and in enhancing both standard of care and adjunctive mitochondrial metabolic therapies as well. If you like videos like this, please like, share, and subscribe. Until next time.